Hello, my dear friends. Today we are going to discuss one simple topic called as can COVID attack for the second time or not? This is Lakshmiya, and we'll have a small discussion about this topic. So, what is meant by reinfection? Reinfection is nothing but in which the viral genome will be infected for the second time. If the infection has been occurred for the second time, if even though the person has been infected with the virus and is capable to produce the antibodies. So once the antibodies are produced in the body, if the infection is happening for the second time, so that can be called as reinfection. So for understanding that, you need to understand one simple topic called as immunity. So what is the immunity? So there are two types of immunity can be seen. Number one is antibody mediated immunity. Number two is cell mediated immunity. Antibody mediated immunity is nothing but through the immunoglobulins. So the antibody immunity mediated immunity can be seen through the production of immunoglobulins. Once a virus or a pathogen enters, in response to the virus or pathogen, the immunoglobulins production can be expected. So which recognize the parts of the virus. So once the immunoglobulins are produced, so they will try to recognize the virus or the parts of the virus once if it can able to produce for the second time. Whereas in the case of cell mediated immunity is nothing but through the cells such as T cells and B cells. T stands for thymus cells and B stands for bone marrow cells. So these T and B cells can able to capture the pathogen. This type of immunity is called a cell mediated immunity. So these are the two most important types of immunity. One is antibody mediated immunity and cell mediated immunity. And antibodies could either be binding antibodies or neutralizing antibodies. So here also we can see there are two types of antibodies. One is binding antibodies and neutralizing antibodies. So binding antibodies which bind to the virus and sensitize the immune system, or neutralizing antibodies which directly bind to the virus and interfere with the virus function and will destroy the virus. So these are the two types of antibodies. One is binding antibodies and neutralizing antibodies. Binding antibodies will stimulate the production of the antibodies, whereas in the case of neutralizing antibodies will try to destroy or combat the presence of antibodies. And unlike antibodies induced by SARS-CoV and MERS COVID, so SARS and MERS, severe acute respiratory syndrome and Middle East respiratory syndrome, which have been happened in 2003 and 2012 respectively. So the outbreak has been very much familiar, and the secondary infections are not possible for those things because the antibodies that has been produced in those individuals stayed for more than 1 to 2 years. So because of the presence of the antibodies for more than 1 to 2 years, those individuals are comfortable in a position to fight against the secondary infection or reinfection. But whereas in the case of SARS-CoV-19 infections, demonstrated that the antibodies are not in a position to stay in the human system for not more than 4 to 5 months. So because of this, the secondary infection can be possible. But all the individuals are not in a position to exhibit the secondary infection because most of the commercial antibody tests. So the complication in this is nothing but most of the tests that are being conducted by using the antibodies are binding antibodies. These are not the neutralizing antibodies. So while immunological memory is more accurately reflected by the presence of neutralizing antibodies. So this is the one simple task. Whatever the tests we are conducting, mostly on the basis of binding antibodies. As you know, there are a second type of antibodies called as neutralizing antibodies. These commercial diagnostic tests are not producing the neutralizing antibodies. So that is the one important limitation for the diagnosis of the COVID-19. Secondly, the role of cell-mediated immunity has not been elucidated so far. So, whatever cell-mediated immunity is nothing but the immunity that can be generated through the B cells or T cells has not been identified accurately. So, it must be understood that all the individuals may not be able to get possibility to get the secondary infection. So why? Because their cell-mediated immunity may be very much stronger, and antibodies that are using the diagnosing are mostly the binding antibodies, not the neutralizing antibodies. So because of these particular things, so much more work has supposed to be done. But the possibility of reinfection is there, but not as much as it is expected. So that is why, which is popularly called as second wave. Second wave is evidently possible because of these two important parameters. So the number one is the life of the antibodies that are produced in the human system because of the COVID-19 are just having 4 to 5 months only. So thanks for watching. Meet in another session.